This is the last set of slides for the macromolecule unit. This is the structure and function of macromolecules, specifically talking about proteins and nucleic acids. There are four large classes of biomolecules. We've talked about this: carbohydrates, lipids, proteins, and nucleic acids. They are largely molecules composed of thousands of covalent bonds. Their structure and function is super important. Proteins include a huge diversity of structures and have a ton of functions. Proteins account for more than 50% of the dry mass of most cells, so if we took away all the water. Protein functions include support, storage, transport, cellular communication, movement, defense. All of your antibody activity is going to be proteins. These are just some examples of the functions, and we'll talk in more depth about these. Enzymatic proteins, they accelerate chemical reactions. For example, the enzymes that digest and catalyze your food. Storage, they store amino acids. For example, casein, the protein of milk, stores amino acids for baby mammals, which is why these young organisms are going to consume the milk because of the amino acids that are stored there. Hormonal proteins, any sort of hormones, so insulin, glucagon, all of that stuff, those are proteins. They are signaling molecules. Defensive purposes, the antibodies are proteins that attack certain viruses. Also transport proteins. There are ones that physically move throughout the body, and there are ones that are embedded in membranes and transport proteins. When we think about like sodium potassium channels, that's a protein. Receptor proteins, the proteins that are involved in the synapse and the movement of neurotransmitters through the neurotransmitters, hormones moving through neurotransmitters. This is going to be receptor proteins, and the signaling molecules are also proteins. It'll also provide structural support, so keratin in our hairs, collagen in our skin, any connective tissue, these are all proteins. A little bit more about enzymes, and we will probably do a lab about this because this is super important in the AP curriculum. Enzymes are a type of protein that acts as a catalyst to speed up chemical reactions. Enzymes can perform their function repeatedly, and they do this throughout the process of their whole life. If we look at the bottom right corner, this is the very classic graph of enzyme activity. The red line is what happens without the enzyme, and it requires a huge amount of energy to complete the reaction, whereas the blue line shows that there's only a small amount of energy put in to have the reaction occur. And so what the enzyme actually does is it decreases the activation energy. So it provides a little bit of extra energy so that it does not have to look for those sources elsewhere. Amino acids are an organic molecule with a carboxyl and amino groups. Carboxyl is going to be COOH, and amino groups are going to be NH2. We also see another R group. Amino acids differ in their properties due to the different side chains or R groups that show up. But the amino acid monomers are going to be the single units that create our proteins. Polypeptides are branched polymers built from the same set of 20 amino acids, and so we go from our monomer, which is amino acids, to polypeptides, which is going to be long chains of amino acids, to the actual protein, which is the biological functional molecule that consists of more than one polypeptide. A lot of these nucleic acids are hydrophobic or water-fearing, specifically the R groups or the side chains. And so often what we see is that parts of these are hydrophobic while the rest of them are hydrophilic or water-loving. These are some side chains that are hydrophilic. Do not feel like you need to memorize this. This is just looking at how water or proteins are going to be partially hydrophobic, partially hydrophilic. Then we have hydrophilic but electrically charged R groups, a totally different class of nucleic acids, and they're going to have very specific behaviors as well. These amino acids are linked by peptide bonds to create your polypeptide chains. A polypeptide is a polymer of amino acids. They range in length from one or a few to thousands of monomers, and each has a unique linear structure with a carboxyl end and an amino end. And then everything in between can vary, and all the R groups that are attached can vary as well. This is how a peptide bond forms. It is between the NH2 and the COOH, the carboxyl, and the amino group of two nucleic acids. This is also looking at how peptide bonds are forming. This is a water loss reaction. So we see water is a product where the carbon and the nitrogen end up bonding. We call it a dehydration reaction. When we get to protein structure and function, the first thing is we have a long strand of amino acids bound with peptide bonds that makes our polypeptide chain. Once the string of amino acids starts to interact with itself and its environment, which is often water, so a polar molecule, then we have a functional protein that starts to twist and fold into a very unique shape. And the function actually comes from that unique shape. There are 4 levels of protein structure. Primary is the unique sequence of amino acids. Secondary is going to be coils or folds in a polypeptide chain. Tertiary is determined by how our R groups interact, and if we remember, we have hydrophobic, hydrophilic, and electrically charged R groups, so they're going to interact in a variety of ways. And then my quaternary structure, which is when one protein interacts with another polypeptide chain. A good example of this is hemoglobin. That is actually multiple proteins interacting to make the hemoglobin protein. This is an example of your primary structure. It is determined by genetic information, and it is created from your DNA and then your RNA, which is transcribed into this chain of amino acids. We see that the secondary structure, it starts to coil. There are 2 typical structures, the alpha helix and the beta pleated sheet. The alpha helix is an actual helix. The pleated sheet is the, obviously, it looks like a pleated sheet. Again, we see them here. Tertiary structure is going to vary a ton because we have all these different R groups. Some R groups are going to have ionic bonds or form strong covalent bonds. Some will have intermolecular forces or hydrophobic interactions and repel from each other. It all really depends on what the R groups are. But we see a hydrogen bond forming, we see hydrophobic interactions, we see an ionic bond, a disulfide bridge. It really varies. The quaternary structure results when 2 or more polypeptide chains forms one huge macromolecule. Collagen, the proteins in your skin, actually consist of 3 polypeptide coils that become a rope. Hemoglobin, another example, is a globular protein, so not a long strand, consisting of 4 polypeptides. 2 of them are alpha-shaped and 2 are beta chains. This is kind of a summary. Take a look through it if you need to. When we see a change in one of these 4 structures, we see disease. Sickle cell is a change in the primary structure, which then goes on to affect secondary, tertiary, and quaternary structures. Basically, a single change in the order of amino acids changes how your red blood cells are going to form. Therefore, it forms the sickle shape, and then you see all of the leading up issues. Again, in normal hemoglobin, which is the top, we see that the chain is normal. It forms a beta sheet. It wraps around itself and it joins with other molecules. We see that there's an exposed hydrophobic region on our diseased sickle cell hemoglobin, and therefore it does not bind in the appropriate way, and it shapes into a sickle cell. In addition to primary structure, physical and chemical conditions can impact or especially negatively impact the structure. pH, salt concentrations, temperature can cause a protein to unravel. The most obvious example of this is an egg. The white of the egg contains tons of proteins. When we add heat, we see the structure changing drastically in the form of a color change. It becomes white. We see the proteins are physically becoming denatured. A denatured protein is inactive, and so it cannot function anymore. Something to think about is when someone has heat stroke or some very serious heat issues, one of the things that we worry about is the proteins in the brain starting to denature, and they will not function anymore. The other group of macromolecules that we are addressing is nucleic acids, which store, transmit, and express hereditary information. The amino acid sequence of polypeptides is created by a sequence in our genes. Genes are made of DNA, and nucleic acid made of monomers called nucleotides. So our very smallest units for nucleic acids are nucleotides, and there are two types of nucleic acids that will form. Deoxyribonucleic acid, or DNA, and ribonucleic acid, which is RNA. DNA provides directions for its own replication. DNA then directs synthesis of RNA, specifically messenger RNA, which can leave the nucleus and control protein synthesis, which happens in the ribosomes using tRNA. Again, we see blue is DNA, red is RNA. The mRNA leaves. The mRNA is read by the ribosome, specifically the tRNA, and it makes a polypeptide chain of single amino acids. Each nucleic acid, so your DNA and your RNA, is made up of monomers called nucleotides. These consist of a nitrogenous base, a pentose sugar, and one or more phosphate groups. So again, we see that the nitrogenous base is going to be the rungs or the center of your DNA. The sugar is going to be part of the backbone, and the phosphate is also going to be part of the backbone, which sticks off the edge basically. So this blue, light blue area is the backbone, and we would see that there should be another one running parallel to it if this is DNA. If this is RNA, this is the strand we see. There are two types of sugars, deoxyribose, which we see in DNA, and ribose, which we see in RNA. There are 5 nitrogenous bases, and you probably know them as the letters that make up our DNA and our RNA, the A, G, C, T, and then U in RNA. They are cytosine, thymine, uracil, adenine, and guanine. We need to know the pyrimidines and the purines. Pyrimidines are going to be your cytosine, thymine, and uracil. The easiest way to remember them is they are a single ring, whereas the purines are going to be adenine and guanine, and they are double ring macromolecules. Keep in mind that a pyrimidine is always going to pair up with a purine in our DNA, which is why A and T and G and C pair up, or A and U pair up in RNA. Here we see it. We have our purine paired up with our pyrimidine on every rung. We see we have our sugar phosphate backbone, and this is going to be your double-stranded DNA. The purine and the pyrimidine are bonded using hydrogen bonds, super important, specifically 3 actually. This is, again, kind of covering what we talked about. Pyrimidines have a single 6-membered ring. Purines have a 6-member and a 5-member ring fused together. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose. These adjacent nucleotides are joined by covalent bonds that form between the OH group on the 3-carbon and the phosphate on the 5-carbon. And so these links create a backbone, which is our sugar phosphate backbone, which allows my nitrogenous bases to line up in the center of my DNA and pair up. RNA molecules usually exist as its single chain, whereas DNA molecules are going to be 2 nucleotide strands that form the double helix. They run anti-parallel, which we will talk more about during genetics, but we need to know that it runs 5 end to 3 end, and they run opposite to each other. We'll talk about what that means. It has more to do with when we are reading the DNA. We should know this as well. I covered it. Please pause and read this over if you need to. It is called complementary base pairing. You always have a purine with a pyrimidine. Again, we see it here.